This paper 2 question asks us to outline the uses made by a plant of named carbohydrates. So, let's walk through the main roles carbohydrates play in plant life, using specific examples. So, we will write that plants use carbohydrates in several ways. Glucose is used in respiration to release energy, ATP, needed for various cell activities. Some glucose is converted into starch for storage. Glucose is also used to make amino acids, proteins and lipids for growth and repair. Another carbohydrate, cellulose, is used to build cell walls for strength and support. Additionally, sucrose is used for the transport of sugars around the plant. So, to summarize, plants use carbohydrates in several key ways. Glucose is used to release energy in respiration. Starch stores energy for later use. Glucose also helps build proteins and lipids for growth and repair. Cellulose provides structural support, and sucrose is used to transport sugars throughout the plant. Each of these carbohydrates plays a unique and essential role, showing just how versatile and important they are to the life and function of a plant. Next, there is a 5-mark fill-in-the-blanks question focused on the structure of DNA. It begins by stating that DNA carries genetic information in the form of genes. The task involves completing a paragraph that outlines the structure of a DNA molecule by filling in the most appropriate word or letter in each blank space. The paragraph reads, DNA is composed of the chemical elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. 2 DNA strands twist together to form a double helix. Each strand consists of a chain of nucleotides. The strands are held together by bonds between specific pairs of bases. These bases always pair in a fixed way, T pairs with A, and G pairs with C. The next question states, a student tested a food sample, and the results are shown. The table includes the following tests: the color change after adding Benedict's solution and heating, the color change after adding Biuret reagent, and the color change after adding iodine solution. Based on the test results, the food contains a reducing sugar and protein, but no starch. Therefore, the correct answer is C, protein and reducing sugar only, as it accurately reflects the observations. The next question is based on an experiment involving a partially permeable membrane. A diagram shows a beaker filled with water, and inside it is a membrane bag containing lipase, an enzyme, and cooking oil, a fat. After 30 minutes, tests were conducted on both the contents of the membrane bag and the water in the beaker. To understand what's happening in this experiment, it's important to note that the membrane is partially permeable, meaning small molecules can pass through it, while larger ones like enzymes and fats cannot. Lipase breaks down cooking oil into fatty acids and glycerol. Fatty acids are small, acidic molecules and can diffuse out of the bag into the surrounding water. Based on this, we can predict the test results. Inside the bag, the Biuret test should be positive, purple, since lipase is a protein. In the beaker, fatty acids would lower the pH of the water due to their acidic nature. However, the Biuret test on the water should be negative, blue, as no proteins or fats are expected to pass through the membrane. The table provided in the question shows different test results. Row B correctly matches all predictions. The Biuret test on the water is blue, no protein. The Biuret test on the contents of the bag is purple, protein present, and the pH of the water is acidic, 5.5, due to the presence of fatty acids. Therefore, row B is the correct answer. The next question focuses on enzyme activity in the lock and key model of enzyme action. It explains that fats are broken down by the enzyme lipase into fatty acids and glycerol. According to the lock and key model, the enzyme functions as a lock, with a specific active site, and the substrate acts as a key that fits into that site. For the reaction to occur, the substrate must bind precisely to the enzyme's active site. In this reaction, lipase is the enzyme, and fats are the substrate it acts on. Thus, lipase is the lock, and fats are the key. Based on this understanding, option B is correct, as it correctly identifies lipase as the lock and fats as the key. The next question presents a table showing the results of 4 different food tests carried out on 4 white powders labeled A, B, C, and D. The task is to determine which powder contains a carbohydrate, a fat, and a protein. This one is a bit tricky, but after careful analysis of the results, we observe that the Benedict's test initially turned from green to blue, indicating the presence of some substance. Powder A is identified as the correct answer, as it tests positive for reducing sugars, proteins, and fats. These results align with the criteria provided in the question, confirming that A is the correct choice. The final question in this practice session involves an experiment using a partially permeable membrane and asks what results would be expected from a series of food tests. The diagram shows a beaker filled with water, and inside the beaker is a membrane bag containing a mixture of starch and amylase solution. The setup is left undisturbed for 30 minutes before samples of the water in the beaker are tested with Benedict's reagent, Biuret reagent, and iodine solution. To understand this experiment, it's important to note that the membrane is permeable to small molecules like sugar and water, but not to larger molecules such as starch or protein. Amylase is a protein enzyme that breaks down starch, a large carbohydrate, into maltose, which is a reducing sugar. Over the 30 minutes, the amylase inside the bag will begin converting starch into maltose. Since maltose is small enough to pass through the membrane, it diffuses into the water surrounding the bag. However, the starch and the enzyme amylase, being too large, remain inside the bag. As a result, when the water outside the bag is tested, it will contain maltose but not starch or protein. Based on this, we can predict the following test results. Benedict's test will be positive due to the presence of maltose, showing a red color. The Biuret test will be negative, staying blue, because no protein has diffused out. The iodine test will also be negative, remaining brown, as starch is not present in the water. When we compare these predictions with the answer choices, option B is the correct match. It shows a red result for Benedict's test, blue for Biuret, and brown for iodine, all consistent with the expected outcomes. Therefore, option B is the correct answer. That brings us to the end of Chapter 4. I hope you enjoyed this session and found the video helpful. With this, we've now completed all 4 chapters covered so far. If you'd like to revisit any of the earlier chapters, feel free to explore my channel. Each video includes detailed topical notes either in the description or pinned in the comments section. Thank you for watching, and I'll see you in the next session.